In this practical video tutorial, I'm going to do test for protein in an abnormal urine sample. As a test for protein, we can do two tests. The first one is sulfosalicylic acid test, which indicates whether protein is present or not. And second one is the heat coagulation test, which can indicate whether albumin is present or not. So let's first do sulfosalicylic acid test. To do this test, we have to first pick up clean and dry test tube. In this test tube, we need to take approximately 5 ml of abnormal urine. So I think this is 5 ml, approximately 5 ml of urine sample. In this urine sample, if we examine, it is quite clear. Okay. So as we want to do sulfosalicylic acid test, we have to add half ml of this 30% sulfosalicylic acid reagent. So 8 drops will make half ml. So let's add 8 drops. 1, 2, 3, 4, 5, 6, 7, and 8. After adding it, let's mix it. And as you can see, immediately white turbidity or precipitates are observed. Let me hold it against this dark background. This way you can clearly see this white turbidity or precipitates. This observation of white turbidity or precipitates in case of sulfosalicylic acid indicates that protein is present in this sample. Okay, so when you write down this positive finding in your journal, you need to first write name of test, that is sulfosalicylic acid test. Then under the heading of observation, you will write white precipitates or turbidity is present. And in the heading of inference, you can write protein is present. Okay. If protein is not present, then you will not able to see this white precipitates or turbidity. Now, as a second test, we have to do heat coagulation test. To do this test, again, we have to take one new, clean and dry test tube. Now, we need to fill up this test tube 2/3 with this abnormal urine sample. So, let me add this urine sample. We have to fill it up to 2/3 mark. So, this is a 2/3 full test tube with the urine sample. Now, we need to hold it with this test tube holder because in the next step, we will be heating this urine sample. Now, what we need to do is, we need to heat only this upper part, not the lower part. Okay. Why upper part only? Because when we heat upper part, at that time, we can compare it with the lower part of the solution. So let's do that. So for that, we need a burner. So we will be using this burner for this experiment. And I will hold this test tube at a 45-degree angle. So by holding it at a 45-degree, keep it over a flame and you can move back and forth so that even heating of the upper part of the solution can be done. Momentarily, you might need to remove this test tube from the away from the flame, because if it's overheaten, then it can be boil over and fall outside of the test tube. So back and forth motion, gentle heating, and just remove it for a moment and one more time. So yes, the heating is enough. Let me remove this burner from the field. Okay. So as you can see, the upper part become cloudy, right? It indicates that coagulum is present, coagulum is formed. You can compare it with the lower part. Lower part is still clear and upper part become cloudy because whatever albumin is there, it become coagulated. Now there is one problem in this heat coagulation test. This cloudiness can be given positive also by the phosphate. So there are two substances. One is albumin and second one is the phosphate, which can give this type of observation after heating. So how to differentiate between these two? To differentiate, we can add acetic acid. So we have this 1% acetic acid and we have to add 2 to 3 drops of 1% acetic acid. So let me add it. So 1, 2 and 3. After adding acetic acid, gently swirl it to mix it so that only it mixes in only this upper part only. So as you can see, this cloudiness or this coagulum is getting intensified. So when after adding acetic acid, if coagulum intensifies, it indicates that albumin is present. If phosphate would be there, and would that phosphate had given this cloudiness, then on adding acetic acid, this cloudiness will disappear. But that didn't happen in our case. So in our case, we can say that albumin is present. Now, once again, when you write down in the journal, how you will write? In the under the heading of name of test, write heat coagulation test. Under the heading of observation, write down that coagulum is formed which intensified after adding acetic acid. That entire sentence would be your observation. And under the heading of inference, you will write albumin is present. So I hope that both the test, this sulfosalicylic acid test and heat coagulation test, are clear to you. If you have any doubts or query, please write it down in the comment section below. Thank you.